seen, but today we will see the how to artificial method of vegetative reproduction in plants. There are two type of methods. One is a conventional method and a modern method. Conventional method and a modern method. So what is an artificial method? So that is agriculture farmers and others are also used for a long time cultivation. The peoples are also used in a convenient way. They are called as an artificial method of vegetative reproduction. That is called as a conventional method of reproduction. They are usually done in a farmers, horticulturists also. First is called as a cutting, grafting and layering. Cutting, grafting and layering are the artificial method of reproduction. First one, cutting. So new plants are produced by cutting. This is called a simple method. So when a rose plant in your house, if your friend or relations are asking to you, what happened? So you have to cut a small piece from the plant and that plant keep in a suitable condition that growing into a new one. So this method is called as an cutting method. So that is called as a root cutting, stem cutting and a leaf cutting. So the cut part is placed in a suitable medium. Suitable medium means a suitable condition that is kept in a soil or in other places. That plant is also growing into a new one. So that produces the roots at the region and grow into a new plant. So the example, root cutting malus. Malus means an apple tree. And stem cutting hibiscus, rose, chapota, mango. So these are the examples called as stem cutting. And leaf cutting, yesterday we saw that bryophyllum, that's called as leaf budding or epiphyllous budding, begonia and bryophyllum. These also, young ones are also produced from the leaf. So this called as a plant is cut into many pieces. These pieces are kept in a suitable condition. Each bit develop into a new plant. This is called a simple method of a reproduction is called a cutting method. Then next is called a crafting. So crafting is a technique that is using the two different plants are joined. So this is one plant, this is another plant. These two different varieties are also joined together and they are producing a young one is called as an crafting method. So the plant which is contact with the soil, is called stalk. What's the name? Stalk. So which plant is also growing in the soil, so that part is called as a stalk. And then which plant is used to crafting. So which plant is also used to crafting, this is called as a scion. So this rooted plant we are called as a stalk, and which plant you have to craft or joined to the stalk is called as a scion. That is a desirable plant. So these two plants are also making a small scrap and joined together or with a polythene cover or some mud and carefully tied with each other and leave it for 1 or 2 weeks. What happened? So there is a new buds produced from the scion. So we can detach it from the mother plant. So we can propagate easily. So this method is called as a crafting. So parts of two different plants are also joined together to producing a new plant is called as a crafting. The crafting method is also done in a different methods: bud crafting, approach crafting, tongue crafting, crown crafting and wedge crafting. So we will see one by bud crafting. When you see you take a plant and make a T-like incision in the stalk, T-shaped incision in the stalk, and bark is removed. The scion or bud of scion, T-shaped scion is inserted into the stalk, and when we covered with properly with polythene cover or mud and allowed to for a few weeks, what happen? That producing a new growth from the scion. This technique is called as a bud grafting. And the next one is called as a approach grafting. So here both stalk and scion remain rooted. So both plants, look here, grafting means so which plant is also rooted is called as a stalk, and which plant is used to grafting is called as a scion. But here both plants are also rooted in a approach grafting. So the stalk is grown in a pot. The stalk is also grown in a pot and it brought to the close to the scion. This is called stalk and scion, both are rooted or joined together. So both of them should have the same thickness. So the stalk and scion, both of the stem have a same thickness. A small slice is also opening into the stalk, and both are also joined together and then tied with a polythene cover and leave it for a few 1 to 4 weeks. There's a new branches are also produced from this tip of the plant, and this cutting is called as an approach grafting. And what is a tongue graft? So take a stalk, and this stalk is also cut into obliquely. So the stalk of the plant having this same thickness, and cut into an obliquely. So this the same method. This scion is also cut into obliquely and fixed into the stalk, and covered with a tape or other clay or thread tightly tied with this portion, and leave it for many days. That's producing a new plant from this oblique region. So this is called as an tongue grafting. Next one is a crown grafting. When the large sized plants, large sized plants are also used for grafting, because the stalk is a large in size, but the scions are also cut into wedge shaped and is inserted into the slits and fixed in a proper position, and by using the craft wax. So wax is also fixed to the both stalk and scion. So let's leave it for a few days and develop into a new plant. So this type of technique is called crown grafting. And then what is a wedge grafting? This slit is made. Slit means an opening is made in the stalk, or bark is removed. A twig or piece of a scion inserted into the stalk and tightly bounded with a paper or other things. And when this cambium is also fixed, to the stalk and scion. So here also both this stalk cambium and scion cambium is also, both cambium are also joined together and then develop into a new plant. So this type of techniques we are called as a wedge shaped grafting. And third method is called as a layering. What is layering? So this method is also done in most village peoples. They are propagating a new plant. This is called the easiest method. The stem of the parent plant is allowed to develop roots while still intact. We say in Tamil, layering means padiyam bodudal. So when the parent plant is also, this lower part of the branch is also bent downward to the soil, and this bend downward is also inserted into the soil and leave it for many weeks. The new branches developed and gives us new branches, and we can cut from this region and removed from this part and grown as a new plant. So this technique is called layering. So layering is of two types, mound layering and air layering. So which plant having flexible branches? Flexible means easily bend branches. That lower branches also bend to the ground, and a part of the stem is also buried under the soil and grown as a new plant. This is called as a mound layering. And air layering means when this desired branch is also girdled or removed the bark of these branches at the nodal region, and applying some hormones in this region. That hormone which is help to promote rooting in this region and develop into a new plant. So this condition is called as air layering. So next we will see the modern method of vegetative reproduction. Next is called the modern method of vegetative reproduction. So nowadays the biotechnology, genetic engineers are developed by using the tissue culture. We are producing the large number of plants. So this is a modern method of vegetative reproduction. So this method is also done by a process called as a totipotent. So what is a totipotency? The genetic ability of a plant cell to produce the entire plant. Genetic ability means all the cells are also reproduce their young one under suitable condition. Under suitable condition means sterile condition. Each cells develop into a entire plant. So this process we are called as a totipotency. Next technique is a micropropagation. So the regeneration of a whole plant from the single cell or tissue or small pieces of vegetative structures through tissue culture is called as an micropropagation. So now we are also getting banana and seedless guava. This also produced from the micropropagation techniques. So there is producing the large sized fruits, flowers are also mainly used by the micropropagation or tissue culture techniques. So what are the advantages of modern method? So desired plant can be multiplied rapidly or easily. So desired plants can be multiplied or rapidly. So plants produce genetically identical. So all the plants are also mostly all the plants are also same size fruit, flower, vegetables are also produced from this technique. So those plants do not produce viable seeds. This can be propagated this method. So some plants, for example pineapple, these plants do not producing seeds. This is also easily propagated by this method. And the rare plants and the endangered plants can be propagated this method. So rare and endangered plants can be propagated very easily. So diseased free plants can be produced by meristem culture. So what is a meristem? So meristem is located in the root tip and shoot tip. So this portion is also, meristem is also undergoing division. Again divide and divide gives rise to new cells. So there is a disease free plants are also produced from the meristem culture. So what are the advantages of this modern method? It is a labor intensive and requires skilled laborers. So it's called a very expensive method. And a sterile condition must be maintained for a longer time, which adds to the cost. So this also produced seedlings in a large number of amount or large time, and amount also wasted. And clones are genetically identical. So clones are genetically identical. What are clone? So morphologically, physiologically similar to their parent plant is called as a clone. So all these young ones are also genetically identical. In this modern method, this totipotent, tissue culture, micropropagation, this also adding by the famous scientist called as Stewart, the Cornell University. He selected parent plants from a phloem parenchyma. It's called as phloem parenchyma cells. Phloem parenchyma cells under the culture, they are producing their young one. A single cell develop into a entire plant. So this is a totipotent that is produced by F.C. Stewart, a famous. And then a famous embryologist called Maheshwari, P. Maheshwari. He also a eminent embryologist. He is also producing a large number of economically important plants produced by these techniques. So next topic is sexual reproduction. The fusion of male and female gamete. The fusion of male and female gamete is called fertilization. So which organisms are also involved in this technique? They are called as sexual reproduction. So this sexual reproduction involves what is a flower, parts of flower, what are essential organ, what is pollination, what is fertilization. We will see this chapter. First one, what is a flower? All of you know that flower means a modified condensed shoot is called as a flower. A modified condensed shoot is called as a flower. So what are the parts of the flower? There are 4 bulbs of the flower: calyx, corolla, androecium and gynoecium. These are the parts of the flower. So calyx and corolla, they are not involved in a reproduction. It is known as a non-essential organ of the flowers. But androecium, gynoeciums are involved in a reproduction. They are called as a essential organ of flowers. So this androecium producing the male gamete and gynoecium producing the egg cell. These pollen grains or male gametes reach to the egg cell is called as a fertilization. So before fertilization, there is a large process called as a pollination. So before fertilization, this pollination is occur. Because what is the pollination? Pollination is the transfer of pollen grains from anther to stigma of a flower is called as a pollination. The pollination is followed by the fertilization. The fertilization is fusion of male and female gamete to form a zygote. Zygote develop into fruit. So the fertilization is also divided into 3 headings: free fertilization, fertilization and forced fertilization. So this process is 3 different stages: pre-fertilization, fertilization and post fertilization. What is a free fertilization? So before the union of gametes, the formation of male and female gamete is called as a pre-fertilization. So what are the steps involved in the formation of gamete is called as a pre-fertilization. That means before the fertilization process called pre-fertilization. So the fusion of gamete is called as a fertilization. And then what are the changes occurs after fertilization? That is called as embryo formation, endosperm, formation of fruit, and formation of seeds. These are also developed after the fertilization. So this comes under the forced fertilization. So first one, free fertilization. All of you know that the male reproductive part of a flower is called as an androecium. So androecium consists of number of stamen. Each stamen develop into pollen grain. So the pollen grains are carrying male gamete. The study in your 10th standard, but here we will study it in a detailed manner. How the development of anther, how the formation of pollen grains. So what are the layers are there? How these microspore mother cell develop into microspore? How the male gametes are also formed? How the female gamete egg cell are produced? So we will study it in the later classes. So this tomorrow we will see what is a forced fertilization, how the male gamete and female gametes are also produced from the androecium and gynoecium. We will see the later. Thank you.